this year is very special for us. It's the 75th anniversary of the school. So I have been here only 8 years, so I'm not going to take the credit for the 75 years, but hopefully I'll make it 75 years as well at the school. I'd like that. Anyway, it's a very special year, and during this year we are going to have several events. We hope to have several events that are going to be organized to celebrate. Some of them are going to be more of a lecture series for the public, and some of them are going to be very scientific that we'll organize at different locations. So please keep in touch with us to learn more about the celebration, and hopefully you can all participate in at least some of the events. I want to take this opportunity to thank, first of all, some of the people that are helping us organize this series, as well as help the school sustain during these 75 years. As a research institution in the country, we are competing for national grants, so we get a lot of support for the research that we do from federal organizations, state organizations, a little bit of the local community. However, a lot of the activities that we do are in fact not supported by the general organization of the research in this country, and we rely heavily on the philanthropy and the gift that are being made to the school. We count on that, and we hope that any of you interested to support the school and help us for another 75 years will contact us. I'd love to hear from you. If you have any interest in supporting the school, please contact me either today or at a later time. We would really appreciate that. In the meantime, I want to take this opportunity to thank the Bank of America that joined us this year as a presenting sponsor for the series. We appreciate their support of our series and the University of Miami. We want also to support another bank, the First National Bank of South Miami. And then we have individuals that are contributing, and that includes Bill Galway, who is here with us this evening, Cheryl Gold, who is also a key player in the organization of this series in terms of selecting the speakers and participating in general, as well as supporting financially the series. So thanks, Cheryl. We really appreciate your help. We have a support from the Shepard Broad Foundation, the Key Biscayne Community Foundation, the Joanne McCoffin Foundation, Melissa and Taylor White, Mary Lou and John Dasberg, the Wang family, and I have seen them in the audience, and the Southern Glazers Wine and Spirit. So thanks to everybody. And again, as I have emphasized, a lot of what we can achieve as a school relies on philanthropy. That includes, not least, but very important scholarships for our students, as well as an endowment for some of the professors that allows us to recruit the top professors in the nation. Over the past years, we have started, as part of the series, to introduce also one of the alumni of the school that we consider a very successful alumni due to what they do and their contribution to society and to the environment in general. And this evening, I am delighted to introduce Laura Bracken. Laura has been with the university for a little while. I'm not going to say how many years because she still looks like she's 16 years old. So you're not going to be able to say anything about that. But Laura received her master's from our Marine Affairs and Policy in 2007, so that was a while back. And since then, she has been working in our advancement office, and now as the outreach coordinator for one of the major programs that we have at the school. So Laura, if you wish to come and say a few words.
Hello, thank you, Dean Avasar. As he said, I'm Laura Bracken. I am originally from Pennsylvania, but I came to Miami for my master's in Marine Affairs and Policy, specifically because Rasmus offers a very interdisciplinary program. I was able to take classes, sort of skills-based classes like GIS and field course in Belize and physical oceanography and marine policy, and sort of tailored it to what I wanted to do and what I felt was important for me. So that really is what drew me to Miami. Fortunately, after I graduated, I was hired full-time, and I've actually been here for 10 years. I just had my 10-year anniversary with the school, so I'll admit how long I've been here. Thank you. Yeah, so I've been full-time for 10 years, and my first 5 or 6 years was in advancement as outreach manager for the Rosenstiel School, so I know a lot of you already through events like Sea Secrets. I'm coordinating programs like that. Now I'm the program manager and outreach manager for CARTH, which is one of 2 major oil spill research consortia based here at the University of Miami. The other one you will learn about in just a few minutes. But CARTH studies ocean currents and how oil gets transported. And it's sometimes very complex, and as an outreach manager, I was sort of struggling of how to engage the public and how to get the community involved in rather complex research. And luckily, the stars aligned, and through partnerships and networks that I have in South Florida, I helped to create the Biscayne Bay Drift Card study. We'd call it Bay Drift for short. And we release these little drift cards into the bay, and they float along with the currents, and people find them, and there's instructions, and they report them to us. So it's very simple, but it's a really great community collaboration. It's a citizen science project, so if you want to get involved, just Google Bay Drift, and you'll find us. And it's been a really great way to sort of bridge the scientific community with the local community here and give back. We're learning a lot of really great information about our local ecosystem. Even though most of our research is based in the Gulf of Mexico, we wanted to provide some information here locally as well. So it's been a really, really great experience. And I just want to end saying that one of my favorite aspects of being a part of the Rosenstiel School community is just constantly being surrounded by the amazing scientists we have here. And every day for the last 12, 13 years, I've been learning from all of these people and being exposed to cutting-edge research. And for me, it's exciting, and I love being a part of it. But I also think it's so great that you all get to be a part of it too through events like Sea Secrets. So I'm thrilled to be here tonight with you, and I hope to see you at many more Sea Secrets and other events throughout the year. So thank you. Have a great night.
My great pleasure to introduce this evening one of our fantastic colleagues at the school, Professor Martin Grosell. Professor Martin Grosell is one of the lead PI, one of the 2 lead PIs that capitalized on the funds that were distributed by BP after the oil spill in the Gulf. And the 2 projects, by the way, we are the only institutions in the country that received the 2 projects from BP to work on that, and that just emphasizes the type of activity that is going on here and the quality of research that is going on. The project that Martin leads is called, the acronym is called RECOVER, and it stands for Relationship of Effects of Cardiac Outcomes in Fish for Validation of Ecological Risk. I have covered pretty much 90% of your talk, Martin, right, with that. Dr. Grosell is our METAC professor of ichthyology with a specialty in environmental physiology and toxicology here at Rosenstiel. He has received a master's degree and a PhD in 93 and 97 from the University of Copenhagen in Denmark. And he has been with the school since 2002. He leads one of the biggest, largest, I would say, research program at the school with a total of about 12 to 15 postdocs and graduate students. He has published extensively, a very prolific writer with more than 200 peer-reviewed papers, which is very, very impressive. So without any further delay, please help me and join me in welcoming Professor Martin Grosell.
Thank you very much. And Ronnie, thanks for the kind introduction, and thanks to all of you for showing up, and also the ones that are in the adjacent room at the wet lab, as we call it. It's really been a ride. It's been a pleasure being here, and I'm delighted and humbled to see this many people here interested in what we're doing.
You have information now that this rig has gone under?
It has gone under the surface at about 10:21 this morning. Coast Guard got word that the vessel has gone under.
What's the implication now of that having gone under? We've seen it burn, which means some petroleum was burning, natural gas or oil. At this point, is that oil in danger now of escaping into the sea?
That is correct. We've got the potential right now is you got almost about 336,000 gallons of sweet light crude oil per day that was coming out of that well. So that oil will be, released into the water now. So the priority is working with our partners in this, especially BP, and getting the environmental part of this underway. Coast Guard is still searching for those 11 missing crew members as well.
I thought I'd take this moment to remind everybody what happened in 2010, April 20th to be exact. And many of you probably know exactly where you were. You remember this clearly. I know I do. The 11 people that the Coast Guard were looking for, we all know now, they ended up being fatalities, unfortunately. You probably also remember being frustrated every day after this announcement to hear that the well was not capped, oil was still flowing into the Gulf of Mexico. I know I was. It took about 87 days. It resulted in 3 to 5 million barrels of oil spilled into the Gulf of Mexico. That makes this spill the largest peacetime spill in the history ever. This is a massive spill. It resulted in 68,000 square miles of ocean directly impacted and covered about 125 miles of coastline and about 10 times as much shoreline because of all the creeks and indentations and points and stuff along the Gulf Coast. So this is a massive coastal area that was impacted by the spill, and of course also an area in the open ocean. Like the nearshore impacts were visible and they were pronounced. It's been estimated that up to 8 billion oysters were lost. That's equivalent to 500 million pounds of fresh oyster meat over 3 generations that we assume it takes these oysters to recover from the spill. That's a lot of oysters. Up to 80,000 individual birds died directly as a result of the oil spill. I'm sorry, it's going to get better later, I promise. 5,000 large turtles or larger juveniles were killed by the oil spill, and over maybe 150,000 juvenile sea turtles passed directly due to this oil spill. And we also had impacts on various marine mammals. Bear terrier bay bottlenose dolphins suffered 35% excess mortality and 45% excess failed pregnancies. If we assume no active restoration, this population will take close to 40 years to recover. So I hope what this is making clear is that this was an epic environmental disaster. It's no joke. Now, I mentioned the open ocean also. There's been a lot of focus on what happened in nearshore environments and of course along the north coast of the northern Gulf of Mexico. But in fact, most of the oil remained at sea, and that's actually where I believe most of the important impacts were. You've all heard that oil and water doesn't mix, but unfortunately that's not entirely true. What happens when you add oil to water, of course, is you form a sheen on the surface. These are the sheens that will cover birds and will wash ashore and soil the beaches and have many other visible and direct immediate impacts. But in addition to that, compounds from the oil will leach into the water and become solubilized. There are many, many hundreds of these compounds. The ones we're most concerned about from a toxicology perspective are referred to as polycyclic aromatic hydrocarbons. And you see some of them here. Naphthalene is a low molecular weight. Phenanthrene is an example of a 3-ringed or middle molecular weight. And then pyrene is one of the heavier PAHs that we find in water that's been contaminated with oil. This is just a snapshot of the 50 most common PAHs found in water that's been contaminated with crude oil from the Gulf of Mexico. And you can see that the 3-ring PAHs in purple here are the most abundant. And that's a concern because those are in fact the ones that are most toxic to fish and other aquatic organisms. Now, measurements from the Gulf of Mexico during and after the spill in the impacted area, depending on how you evaluate this and what samples you consider, I think it's safe to say that an average concentration during these 87 days in the northern Gulf of Mexico in the impacted area was around 20 micrograms of total PAHs per liter. Now, this number will not mean a lot to you at this point, but I want you to keep that in mind as I go through this presentation, because I'll be talking about concentrations that elicit effects on marine organisms. And it's important to keep this as a reference, and I'll remind you as I go through. Now, with this in mind, I think there's good reason to be concerned about animals in the northern Gulf of Mexico, and in particular, pelagic fish. So these are large fish that live in the open ocean. And why should we care about them? Well, I want to show you some data here. This is published back in 2010 out of Barbara Bloch's group. And what it's showing on the left panel here is the abundance of bluefin tuna, those are the filled symbols, and yellowfin tuna and the oval symbols over time in the Gulf of Mexico. And you can see that bluefin tuna, they show up in the spring and they stay through late spring, early summer, and then they leave the Gulf of Mexico again. They actually come in the Gulf of Mexico to spawn, and they spawn in the northern part of the Mexican Gulf, or Gulf of Mexico. And you can see the yellowfin tuna, they're present year-round, highest in abundance during the summer. And if you look at the bottom left of the slide here, you can see that I've indicated the timing of the spawning event for bluefin and yellowfin tuna. So the abundance of these animals and the timing of the spawning is occurring at the time of the oil spill. On the two right-hand panels, you see in the top bluefin tuna and the bottom yellowfin tuna, and this is sort of the location or the distribution of these animals that are caught and reported on the left side. And again, you can see in the northern Gulf of Mexico, and I'm going to use the pointer here because that's going to show up in the next door room as well. This is the area that's directly impacted by the spill. You can see that we have lots of bluefin tuna in that area spawning at this time, and the same is definitely true for the yellowfin tuna. So the take-home message here is that these large pelagics were present at the time of the oil spill, and they were spawning at the time of the oil spill. It means that not only were the adults, but also the very sensitive early life stages directly impacted by the oil spill and exposed to the PAHs I just discussed. In the fall of 2010, we were approached here at Rasmus by NOAA to assist with assessing the impact of these pelagics resulting from the oil spill, and we were very excited to do that. And a few years later, we were able to publish the first report of our efforts in 2014 in PNAS. And what we're showing in this study is that these large pelagic, in this case, it's bluefin tuna on top and yellowfin tuna at the bottom, showed severe impacts at concentrations as low as a few micrograms of total PAHs per liter. I'm showing the exposure concentrations here. And again, remember the reference that I mentioned earlier at 20 micrograms per liter. And then take a look at these fish. If you look at the fish on the right-hand side, those are the impacted fish compared to the control fish on the left-hand side. These are not happy fish embryos, and in fact, they're not going to live but another day or two, and then that's the end of the rope for them. Okay? So there definitely was an impact by this oil spill on these large pelagics. Now, bluefin tuna and yellowfin tuna are extremely difficult to work with. The work on the bluefin tuna was done in Australia, and the work on yellowfin tuna was done in Panama. We did that in Panama in 2011. So we wanted to look for additional models to look at impacts on pelagic fish in the Gulf of Mexico. And we're fortunate to have, this is an aerial view of where we are right now. We are sitting in this auditorium right here, and there's people in the building over here as well, the overflow. And the experimental hatchery is right across the street right here, conveniently located right next to the water. And in that facility, we are able to raise, at this point, not bluefin or yellowfin tuna, but mahi. And I'm just going to take you through about a minute of a video that sort of illustrates how we do that. We typically interact with local fishing charters. We go offshore. We only have to go out a few miles here to hook and line young adult, sexually mature mahi-mahi. We bring them on board and maintain them alive in this holding well that you see on board the vessel here. People think that I do this every day. That's not true. This happens a few times a year. But it is fun when we get to do this. We can maintain these fish throughout the day on board this research vessel. We then pull the boat up on shore. This is right across the street here by the hatchery. We carefully carry these fish individually into these tanks. These tanks are quarantine tanks. And the good thing about mahi is they start feeding right away. They feed within a day after captivity, and usually within about a week they start spawning. Those are fertilized embryos. And this offers the advantage that we can look at these embryos in all life stages. These are larval fish, juveniles. Here we have F1s, young adults, that we can do various experiments on. This is an experiment on a fish in a swim tunnel, and I'll touch on that later. Now, the ability to study these animals at all life stages gave us a huge advantage trying to assess the impact of the oil spill on marine pelagics from the Gulf of Mexico. What I'm showing here on the left is a control mahi. This is 48 hours post-fertilization, so it's newly hatched. And what you can see if you look carefully there is you can see the heart. The heart is beating happily. This is what a happy and healthy young mahi looks like. Okay? I'm going to now start the video next to it, which is an oil-exposed animal at the same age from the same batch. And you can see here with the naked eye that the heart rate is significantly lower. And you can also see, if you look at the flow around the heart here, that there's definitely some disruptions. This is exactly what we saw in the bluefin tuna and the yellowfin tuna. So the bottom line here is that the mahi is actually an excellent model to study impacts of oil spill on marine pelagics. And it's a lot easier, although it's not easy, but it's certainly easier than the large pelagic tunas, for example. This is a summary of some of the impacts we see on embryos and larvae from mahi. 96 hours of exposure will kill them at concentrations less than 6 micrograms per liter. Again, remember, in the Gulf of Mexico during the spill, we saw over 20 micrograms per liter. The cardiac morphology is impacted at low concentrations. Heart rate's a little more robust, so is the stroke volume, but the cardiac output, this is the amount of blood that the heart is pumping into circulation, is reduced as concentrations as low as 3.5 micrograms per liter. Okay? So this is bad, and it's about to get worse. What we and others discovered is that while we see a lot of toxicity to this crude oil when we expose them under artificial light conditions like here, it's even worse when we expose them to natural light. Because the UV waves in natural light actually potentiates the toxicity of PAHs. So what you're looking at here is control mortality in animals that have been exposed to oil under artificial light conditions, and you see about 6 micrograms per liter will kill 50% of them. Whereas if we expose them to natural light, which contains the UV, we see that there's about close to a 10-fold potentiation in toxicity. So all the toxicity that I've shown you up until now, you have to multiply that by about a factor of 10. So we see the impacts at concentrations down at about 1 microgram per liter or less. In the Gulf of Mexico during the spill, we measured 20. Okay. Now, based on our data and also data of others that looked at coastal species, NOAA produced this estimate of the total damage to fish in the Gulf of Mexico as an outcome of the Deepwater Horizon oil spill. And I think one number to pay attention to here is 2 to 5 trillion individual fish died as a direct result of this oil spill. That's substantial. If you think of that for a second, that's a lot. Now this number, along with the numbers I showed you for the very unfortunate birds and turtles and mammals, formed the basis for, well, it was supposed to form the basis for litigation, but I think fortunately litigation was avoided and a settlement was made between the government and BP. BP is estimating the total cost of this oil spill to be $62 billion. Ironically, it's only $44 billion after tax. I don't really know how you justify that as a... No, this is no joke, this is how it is. Regardless, that expense to BP includes close to $5 billion in fines and penalties and almost $20 billion for the natural resource damage, which is what I've just discussed. Most of that money is now distributed to the Gulf states in the Restore Act, and funds for restoration are now available throughout the impacted Gulf states. So aside from this tax benefit that BP is getting, which really rubs me the wrong way, I think it was very exciting to be part of this process where you're doing science that very, very clearly matters to a lot of people. To be honest, that's not always the case. Some of the science we do, in reality, maybe 100 colleagues across the world are really excited about it. But what I learned when I did this work is that a lot of people actually care, and that was tremendously satisfying. I will say, though, that working on this process, which was initially intended to build litigation against BP, sometimes was not satisfying because the focus was litigation and not necessarily scientific discovery. And those two things are not always parallel. And for that reason, we were very delighted to have access to the Gulf of Mexico Research Initiative, or GOMRI. In 2014, I put together a research consortium, and we successfully competed for funding from the Gulf of Mexico, the research initiative, to pursue some of the questions that we have unraveled during the initial NRDA assessment that we worked for with NOAA. And I want to say that BP provided these $500 million shortly after the spill in a good faith effort to provide funds for research to try and understand the impacts and hopefully mitigate some of the impacts. But the money is managed completely independently and is subject to an independent peer review process. So BP has no say in what we do when we receive funding from Montgomery, which I think is really, really important. So what I want to do next is I want to move on beyond natural research damage assessment. I want to talk about what we do in our research consortium. Ronnie introduced this earlier as Recover. And very briefly, I just want to say that the hallmark of Recover is that we look at one pelagic and one coastal species. We look at mahi and the red drum. And we consider what we call different levels of biological organization, all the way from the molecular, subcellular, cellular, all the way through population level impacts. And that's what you see indicated in this arrow here. I'm not going to talk about molecular endpoints today or subcellular endpoints. I'll talk largely about organ function and intact animals and behavior, leading into discussions about population level impacts. I will touch briefly on cell function at some point. Now, before I get started, of course, I need to formally acknowledge our funding source, Gulf of Mexico Research Initiative. And you're going to have to bear with me. It's going to take a little while for these credits to roll through because over 100 people have contributed to the work I'm going to be talking about today. And obviously, I don't want to leave any of them out. And in fact, another satisfying aspect of this work is the tremendous amount of people that I've been fortunate enough to work with during the process leading up to this particular moment here. Will be done in just a few seconds. Then what I'm going to do after that is I'm going to give you 3 examples of things we've discovered that sort of goes beyond the natural damage or natural resource damage assessment. And 3 examples of things that I think are significant and that were not considered initially as impacts from the Gulf of Mexico oil spill. Like a very common relationship for toxicology is this relationship between size and sensitivity. Typically, the larger the animal, the less sensitive it's going to be to environmental insult. Conversely, small animals are very, very sensitive. And so it's no surprise that during the NRDA process, we looked a lot at the embryos and the early life stages. However, what we've discovered is that even larger animals are highly sensitive to crude oil exposure. And I'll give you an example of that. This here is a mahi that's raised at the hatchery across the street. It's in a swim tunnel. In this swim tunnel, we can dictate the flow rate and thereby control the velocity that this fish is swimming at. Fish tend to maintain position in any on-head current. And by turning up the current, we can turn up the exercise intensity of this animal. So this is exactly like you guys being on a treadmill. I'm not going to demonstrate that here tonight, but with the fish, this is what we do. In addition to controlling and manipulating the exercise intensity of this animal, we're also measuring the metabolic rate. We can do that by looking at the oxygen depletion in the water over time. The type of data we get from this is illustrated here. This is actually not real data, but this is the only data I've made up for tonight. Okay? What you're looking at here is the metabolic rate as a function of the exercise intensity. And no surprise, the harder you exercise, the higher your energy consumption. Right? And one thing we can get from this relationship is when we get to the maximal swimming velocity, and this is maximal sustained swimming velocity. We refer to that as eucrit. This is as fast as these fish can swim for 20 minutes. We can extrapolate up and we can get the maximal energy that these guys can burn, maximal oxygen uptake. In addition to that, we can extrapolate back down to zero activity. And this is really important because you cannot tell a mahi to not do anything. In fact, mahi have to move at all times to ventilate their gills. They cannot not swim. So getting a measure of their true resting metabolic rate is impossible. You have to extrapolate. So that's what we've done here. And the difference, of course, between the minimal metabolic rate and the maximal metabolic rate is what we refer to as metabolic scope or aerobic scope. This is the energy this animal has available for, say, migrations, foraging, avoiding predation, spawning, all these really, really important events for surviving and maintaining a population in the environment. So let's look at eucrit and metabolic scope in animals that have been exposed to oil. This data here on the left shows the metabolic rate of animals, control animals exposed to just over 2 micrograms per liter and animals exposed to 8 micrograms per liter. And you can see that there's a progressive decline that is significant at 8 micrograms per liter. So these guys have a reduced metabolic scope when they've been exposed to 8 micrograms per liter for just 24 hours. Again, remember that the oil spill lasted for 87 days and the concentrations in the environment were over 20 micrograms per liter. The swim performance is impacted. There's no surprise there. If you have a reduced metabolic scope, you're not going to run as fast on a treadmill. That's just how that goes. So these animals don't swim as well. And as you can probably imagine out there where it's a matter of either eat or get eaten, this is a severe disadvantage. And we're curious about what is causing this impact. It can really be 2 things. It can be the gills that are impacted or it can be the heart that's impacted. We looked at the gills. The gills are fine in these animals. and that turned our attention to the heart. And what you have here is the heart of an F1 mahi. It's about 90 days old. It was raised at the hatchery, and it's alive. It's anesthetized, but this is open heart surgery on a fish. And what we've done here is we've placed a transonic flow probe around the blood vessel that leads from the heart to the gills. We refer to that as the ventral aorta. And by having that flow probe there, we can measure the heart rate of this animal and also the amount of blood that leaves the heart for each contraction. Okay? When we do that, what we see is that the heart rate is not impacted. That's the top left panel. The top right panel is the stroke volume. This is the amount of blood that leaves the heart for every contraction. We can see that's reduced by over 40%. This is a massive reduction, massive reduction. And of course, heart rate times stroke volume equals the cardiac output. That is the amount of blood that the heart delivers to circulation continuously in this animal. And again, that's reduced by about 40%. If you had a reduced cardiac output by 40%, you got on a treadmill, you would fall off right away. And that's why these animals can't swim. We went one step further here and tried to look at whether we could explain this at the cellular level. And this video here shows an isolated cell from the heart of a mahi. This is referred to as a myocyte. You see the cells are very long. And it's stimulated electrically just like it is in the heart. And whenever there's a stimulation, you see there's a contraction of this cell. And what we've shown is that these myocytes, when they're exposed to oil, these heart cells, they are less able to contract. So each individual cell in the heart has a reduced ability to contract, which means that the heart has a reduced ability to contract, which means that the heart cannot deliver as much blood into circulation. This is why these fish do not swim as well, and this is why they have a reduced metabolic scope. Okay. Now the question, of course, is, does that really matter? I've not discussed the molecular initiating events. We do know what's going on at the molecular level to cause this, but I have shown you now that we have this reduced shortening of the sarcomeres in the shell that leads to reduced shortening of the cell. This leads to the reduced stroke volume and cardiac output. And we know that at the intact animal, this leads to reduced aerobic scope or metabolic scope, and also a reduction in swim performance. So these animals swim less well. And the question, of course, is, what does that do to populations in the wild? Will that have implications for migration? Will it limit migration? Will it impact foraging activity or spawning activity in the wild? And I'm going to address that towards the end of my presentation. I'll get back to that. I now want to turn my attention to a single example of things we discovered after the NRDA, things that I believe is very important, but was not considered by the NRDA. We have noticed that there's an impact on vision in juvenile fish exposed to oil. These are just stills from the videos I showed earlier. And if you look carefully at these, we're focused on the heart when we're looking at these. But if you look at the eye here, you see a clear difference. The animal on the left is the control unexposed animal, and the animal on the right is an oil-exposed animal. You see the eye is smaller and it's less distinct. Okay. Now let's go back and look at the first study we published in PNAS in 2014. You see exactly the same on both the bluefin tuna and the yellowfin tuna. There's a tendency for a reduced development on the eye. Going back through the earlier literature, looking at images of fish that were addressing, say, heart function, for example, we noticed the same thing. So we're not the first to report this. However, many of the earlier scientists did not notice it. They were looking for something else. They did not see the heart. In addition to that, we actually have gene expression data showing that, or indicating that the development of the eye and the development of the ability to see is impacted by oil exposure. So of course we're curious now, can we do a vision test on these fish embryos or fish larvae to see whether vision is indeed impaired? And it turns out you can. This is a cartoon because it's a little easier to illustrate on a cartoon. This is the equivalent of you going to your physician and having a regular eye test, reading the letters on the wall. What we do is we place a fish in a cylinder with alternating black and white stripes, and we then rotate that cylinder. Don't ask me why, but for some reason, fish tend to track these stripes. And they can do that at increasing speeds until a point where the speed is now so fast that these stripes can no longer be resolved by the eye of this animal. So now it's just a blur they can't keep up anymore. Okay? We can then turn the speed counterclockwise, we can repeat the measurement on the same animal, and we can do that on oil-exposed animals and naive animals that have not seen crude oil. Next, I'm going to show you what this looks like in real life. You can see the fish at the bottom part. Let's see here. This is the larval mahi right here. And I'm going to start the video now, and you can see a real experiment. You can see this fish is nicely tracking the stripes along the side here. We can then increase the speed, and that's what we're doing. And I'm going to switch to slow motion in a second, so you can just enjoy how perfectly this animal will track the stripes. This obviously is not an oil-exposed animal. Okay. By gradually increasing the speed, we can determine the threshold at which these animals can no longer differentiate the black and the white, and thereby get an indication of their vision, or visual acuity, if you like. And this is what the data looks like. The open bars here are control animals, 7, 8, 9, and 10 days after hatch. So these are very, very young fish. And you can see that the vision develops in these animals gradually as they mature. And then in the gray bars, what you see here is an animal that was exposed to less than 1 microgram per liter for just 48 hours during embryonic development, 5 days prior to this test. And you see that their visual acuity is significantly impaired by oil exposure. Okay? Now, this doesn't kill them, certainly not under hatchery conditions, because there's plenty of light and plenty of food, but in the wild, this could have severe implications for their ability to capture prey. So to now sum up again here, we actually do have indications from molecular studies that peripheral nervous system, so that's the sensory nervous system, is impacted, including the eye. We know that they have smaller and poorly developed eyes when we look at them. I've just shown you now an example of these animals having reduced visual acuity. We have seen, not on the mahi, but on the red drum, we haven't done on the mahi yet, that they have reduced prey capture ability. We can't prove that that's because they can't see the prey, but it seems like a likely explanation. And of course, at this point, we don't know what that does to animals in the wild. Now, a third and final example I want to give you on impacts that occur in these animals that we've discovered since we started the Gomery-funded research is the impact of oil exposure on embryos. We actually know that the metabolic demand and behavior of these embryos change after oil exposure. Okay? Now, the top image here is an embryo about 24 hours after fertilization. Here you have it about 8 hours later, and another 4 hours later they hatch. Very, very fast development. Okay? During this period here, we've measured the metabolic rate, and that's what we're showing on the graph here. The metabolic rate here for control animal is very, very low all the way up until just prior to hatch. Just prior to hatch, a number of metabolic processes kick in and greatly elevates the metabolic demand or their metabolic rate. This is normal. This is what mahi will do under normal circumstances. If we expose them to oil for 24 hours before we start these measurements, this is what we see. We see these 4 consecutive trials of elevated metabolic rates compared to the controls. Now, there's a metabolic demand apparently dealing with these crude oil exposures. And it's unfortunate for these embryos because these embryos are not feeding exogenously. They have a yolk sac that provides the energy for development up until a few days after hatch. So the question, of course, became, does this lead to depletion of their energy stores? And the answer, not surprisingly, was yes. What you're looking at here is a control and oil-exposed animal to the right. This is the control animal. You can see that the entire egg here is filled with yolk. Whereas if you look at the oil-exposed animals, you can see a clear depletion of the yolk area. That's what we've quantified on the graph here. Okay? So they do run out of energy. They certainly have reduced energy, which could be a problem for later survival of these early life stages. Okay. The next slide I'm going to show is a little complicated, so please bear with me. The blue line here shows the buoyancy of control embryos. If they have a score of 1, they're floating at the surface. If they are at 0, they're neutrally buoyant, and if they're less than 0, they sink. Okay? So what you're seeing here is that control embryos for mahi in artificial light, they're floating, and then just prior to hatch, they start sinking, and then you have the hatch rate coming up here. So this is the normal situation for a mahi embryo. If they've been exposed to oil, that's the green line, you see that they're no longer positively buoyant. They're not floating, in fact, they're sinking. And they sink all the way, they're basically sinking all the way through to hatch. If they're exposed to the combination of UV light and oil, this effect is even more pronounced. And UV light in itself also appears to cause a negative buoyancy in these animals. Okay? Now, is that a big deal? Does that matter? And we very much think it does. And it has to do with the fact that ocean currents will differ with depth. Presumably, this normal behavior of being possibly buoyant and floating and then sinking later will position the embryos in an optimal prey-predator field when they hatch. They're going to be moving with the ocean currents. And what we're going to do to try and assess this is really hard to measure in vivo, of course. So what we're going to do is we're going to model the behavior of these embryos in response to ocean currents and oil exposure. And I'm really happy to have one of my colleagues help me with this. He's actually here in the audience today. It's right here. Hi, Claire. So Claire is an expert in modeling ocean currents, and with her models, what we can do is we can actually determine the fate of these embryos depending on whether they're positively buoyant or negatively buoyant. And we can then overlay that with what we know about what happened to the oil concentrations in the Gulf of Mexico during and after the spill. This is going to give us a couple of things. It's going to give us, one, a more accurate assessment of the impact during the spill in the Gulf of Mexico, and two, it's going to give us a much better understanding of the dispersal of these embryos and larvae after spawning in a pelagic fish like the mahi-mahi. So we're excited about that. And I should say this model runs in 3D. You're just looking at the top 1 meter of the ocean in this particular simulation here. So those are 3 examples of things that occurred in response to oil exposure that were not captured by the NRGA assessment. These are not immediately lethal, but I will argue that they have impacts on the population of these wild fish during and after an oil spill. The first thing I showed you was that cardiac performance was impacted in adults. I then showed you that sensory function, we looked at visual acuity, is impacted in larval stages, and this is after only brief exposures during embryonic development at very, very low levels. Okay? So you can survive that exposure as an embryo, but you're going to grow up and live the rest of your life impaired. And that is if you can avoid predation under that impaired state. And then we also showed that embryo energy balance and buoyancy control is impacted. And I would argue that that has implications for both dispersal and survival in the wild. Okay. What I want to do now, or next, sort of towards wrapping up here, is I want to give you 2 examples of some wonderful things we discovered that had nothing to do with the damage of the oil spill. And I promised you I would finish on a higher note, and that's what I'm going to try and do. So it's been pretty gloom so far, I realize that. But actually, one of the wonderful things about the work we've been able to do and we've been allowed to do by the GOMRI funding is the pursuit of science, and the impact of oil, of course, has been the main focus. But every time we do an experiment on mahi to look at oil impacts, we're also including, of course, non-exposed animals. We need that as a reference. And there's nothing or very little known about mahi biology and physiology. So every time we do that and we look at our control animals, we learn very, very valuable lessons about the biology of this animal in the northern Gulf of Mexico. And in fact, I should say, what we've learned about mahi probably applies to all the pelagics, or most of them. And I should also say that mahi are not just present in the Gulf of Mexico. Of course, they're actually circumglobal in both subtropical and tropical waters. They're everywhere. So what we're learning here is important for a lot of fish and a lot of areas in the world. I want to return to the embryos and this issue of buoyancy control. Again here, the blue line is the controls. You see that they're floating up until just before hatch where they start sinking. That's what they're supposed to. This is great. And then we have animals in the red here that have been exposed to UV basically from about 24 hours post-fertilization and then throughout hatch. And you can see that the UV light, which is a component of natural light, of course, causes these animals to be negatively buoyant. Okay? Now, this raises a question, are these embryos actually floating in the surface in the wild? This has been the assumptions by biological oceanographers for close to a century now. And with what we're seeing here, I think that's something we have to re-examine and look at carefully. And what's perhaps most exciting, I think, is these animals here in the orange symbols, they were exposed to UV up until this point, which explains why they're negatively buoyant. And then the UV exposure was terminated, and at this point they regain positive buoyancy before they become negatively buoyant to hatch. Now think about this for a second here. What we're learning from this is that these embryos are somehow able to sense UV light, which is damaging. This is why we apply sunblock. And they're then able to avoid that UV light by sinking deeper to the ocean. So UV does not penetrate even ocean waters that deep. So by sinking just a little bit, you're going to reduce the UV exposure. So what it looks like is they can sense UV light, which is remarkable. And they then have this behavior of avoiding UV light by sinking. It's a reversible behavior. This is not damage. They can reverse it. They can come back to the surface the minute we terminate the UV exposure. This is remarkable. We've looked a lot at how they control buoyancy, and we know a lot of things now about how they don't do it. We don't know exactly how they do it. We're going to hopefully get to that. We've not yet begun tackling how they're sensing UV light at this very, very early life stage. Remind you again that this is what they look like at this life stage here. They are able to sense UV light and react appropriately. Right? This is more than some of us. We don't apply sunblock when we're supposed to. So this is a pretty big deal that we're very excited about. I now want to return to, I think, what could be the $1 million question. The impacts to cardiovascular physiology of function that I've shown you, the fact that the heart is just not working as well and they can't swim as well, and the fact that sensory function, I showed you visual acuity, we also know that olfaction, or the ability to smell, is impacted. What's the combined effects of all these impairments on animals in the wild? That is the million-dollar question. Does it really matter, right? So the ultimate experiment, of course, would be to do a mesocosm experiment. We take one Gulf of Mexico, we add oil, we take another one, we don't, and we compare them. That's impossible. Mesocosm experiments are done typically in smaller enclosures. Mahi are not suited for small enclosures, you just can't do it. So the ultimate experiment, of course, is to go into the Gulf of Mexico, capture wild mahi-mahi, expose them to oil on board a research vessel, tag them with a satellite tag, and then release them back into their natural environment to follow their natural behavior. That's the ultimate experiment. We're doing that in July and August, thanks to GOMRI funding. And what I want to share with you now is some of the preliminary work we did to show that this is indeed feasible. And forgive me, but this is going to be another video. Just another day in the office here. We're out hook and line angling mahi. We're going for slightly bigger fish here than when we are collecting for broodstock. We bring them on board very, very carefully. It's important that these animals survive the handling. We take a little fin clip, that's for DNA identification, and we then fit them with this pop-up satellite tag. And I'll get back to the tag in a second. We hold them on board the research vessel anywhere from 2 to 24 hours. You can see a tagged fish coming around the corner there. This is to allow them to recover from the handling stress, but also later to allow us to expose them to oil on board the research vessel before they're released. We then release the fish alongside these drifters that mark the surface currents. And you'll see these fish in a second swim off here. It was super satisfying to see these tagged fish swim away like this. A lot of juice left in them, very, very strong animals. They actually cope fairly well with the tagging. And that was me trying to catch up with them, which, by the way, is impossible. They're very, very good swimmers. Now, this is what we want to do on oil-exposed and non-exposed fish. These tags are programmed to release after 96 days, where they're going to float to the surface, transmit the data to a satellite, that's then going to transmit the data to us right here. So we know what this animal's been doing for 96 days. How deep has it been? What temperatures has it encountered? Where did it go? And in fact, it's a long story, but we can actually, from these tags, predict spawning events also. So we'll know where they spawn, how deep they go, what temperatures they prefer, these kind of things. And the data is still being analyzed from these preliminary experiments, but I want to show you the trace from the first 3 fish we tagged. We tagged and released them right off Miami here. And what you're seeing here is ocean currents. The warmer the color, the warmer the surface. And what you see, the male moved south. Those are the gray symbols. It lasted for 5 days and then died. It probably did something really stupid. It was a male, so it was consumed by something. The two females went north, as you can see, and they're still going. And in fact, one female lasted for just over a month, and the other lasted for 48 days. We know from the tag that at 48 days, that female was consumed by something. And we know this because the tag reported no light for 16 days after that. It was still moving around, but no light. That means that the tag was inside something else. We also know that that something else was not a mammal because the temperature was fairly cold, so we know it was a fish. So we learn a lot from these tags. And one thing we did learn is that that female actually traveled close to 5,000 kilometers, or just under 100 kilometers a day. Like, imagine swimming 100 kilometers a day, right? Like, that's quite the feat. And then think about what I showed you with the reduction in cardiac performance and the reduction in swim performance. And then ask yourself, can these migrations be achieved if you've been exposed to oil? We don't know, but we'll know in July and August. My guess is that there are going to be some problems for these fish. Now, one thing we can do, which I think is unique, is we can combine the laboratory data that I've shown you some of already with this data that shows the behavior of these animals in the natural habitat to provide some estimates of metabolic demands of wild mahi. What does it take for mahi to stay alive in the wild? So what I've done here is I've broken down the different components of their metabolic requirements. I could have expressed this in calories, which is something that we can all relate to. I've done it here in milligrams of oxygen per kilo per hour because that's what we fish physiologists normally do. But you can replace this with calories. The picture is the same. Staying alive for these animals, ventilating their gills, kicking around, those kind of things, cost about 500 milligrams per kilo per hour. Migration, this is moving these 100 kilometers a day, costs more than that, almost 700 milligrams of oxygen per kilo per hour. Digestion, when they're ingesting a meal that actually costs energy to digest it. You know this from the fatigue you feel after having ingested a meal, right? Especially if it's a big meal. That costs them 240 milligrams of oxygen per kilo per hour. The total metabolic demand adds up to about 1,440 milligrams of oxygen per kilo per hour. Now, we know from the swim tunnel experiments that their maximal metabolic rate is just under 1,700 milligrams of oxygen per kilo per hour. So what this means is that a mahi in the wild is operating at 80% of max, or just under 80% of max, at all times. Like, imagine that. Like, I know we're all busy, but 80% of max all the time. These are animals that are working extremely hard. This is life in the fast lane, and that's one of the reasons why these mahi are so fascinating to work with. And it's also, of course, one of the reasons why they're so hard to work with, which is why we know very little about them. Okay? Now, with some assumptions about food conversion efficiency and energy content of what they're eating, I estimated that a 1 kilo mahi will have to eat 48 grams of prey per day to sustain this metabolic demand. Okay? So I know how much one mahi eats. Now, if I could only figure out how many mahi in the ocean, I can figure out how many mahi or how many prey fish are being eaten by mahi. And I can't figure out how many mahi are in the ocean, but others can, and then they've done that. Now, mahi, as I mentioned earlier, is spread throughout the world. They are in all subtropical and tropical waters, but they do form distinct populations. The mahi found in the Gulf of Mexico, the Caribbean, and the Western Atlantic is one population. And we know that that population accounts for about 25,000 metric tons of mahi in this area. Okay? They eat 48 grams per kilo per day, which means that I can estimate the consumption of these animals to be 1,200 metric tons daily. That is the amount of prey fish that are being consumed by mahi in the wild every day in this area. Okay? This is equivalent to 5% of their body mass, which is a lot. I would have to eat 10 pounds a day to keep up with these guys, which is impossible. And in fact, just as a balance check here, this 5% per body mass per day is about what our mahi in captivity will eat when they're adult and mature. So these calculations are not too far off. This is in fact what they do in captivity also. And so they eat a lot. Why is that important? Well, mahi is not just a top predator, it's actually also prey for billfishes. And billfishes, as you know, are very popular. Very little is known about them, and many of them are threatened in many parts of the world. So the more we know about the basis for their life and their lifestyle and their biology, the better able we are to make decisions about management of billfishes. So this is just, I think, one excellent example of how research dollars that were initially intended to study the impacts of oil have actually done far more than that. We've learned a lot about oil impacts, but we've perhaps learned even more about the basic biology of the creatures that live in the Gulf of Mexico, and we're much better positioned now to understand the ecosystem in the Gulf of Mexico, which is no small feat. And this gets me to the question that I posed in the title of this lecture, which was, is there a silver lining? And it was a really, really bad spill. This was an epic disaster. I don't think anyone can argue with that. But some good came out of it. We now have a much greater insight into what the impacts of oil exposure are. That means that we're much better suited to predict damage and impacts of future spills. And here's a spoiler alert. It's not a matter of whether we're going to have a spill or not in the future. It's a matter of where and when. It's going to happen again. But we're better positioned. We're better ready to deal with that when it does. That also means that we're better able to advise first responders. How are we dealing with this oil spill when it happens? Are we applying dispersants? Are we going to burn the oil? What are we going to do? GOMRI, the 500 million that BP put up initially, so far have trained 544 PhD students and 316 postdocs. I've trained a few of them, but definitely not all of them. Many of them are employed now in academia and in government, and so they are in positions of influence. They are the next generation of scientists that's going to do the science that's going to save us the next time this happens. They're in positions to make decisions about what we do the next time an oil spill happens, or even better, to make decisions about what we do to prevent future oil spills, or at least reduce the risk of them. So this is a clear silver lining. But perhaps most importantly is the immense insight we've gained into the biology and ecology of the Gulf of Mexico since 2010 when we began this kind of work. And we're actually still learning. And this image here signifies that I'm about done. But before I take your questions, I want to draw your attention to different avenues for additional information. We have a website. It's miami.edu/recover. Go there. You can learn a lot more about what we do. You can email me if you like. I'll do my best to get back to you ASAP. Of course, we have a Facebook page. We're on Twitter. We have a YouTube channel. So please feel free to explore that for additional information. And with that, I'm delighted to take any questions you may have. Thank you for that question. I'm going to repeat the question so everybody can hear also in the adjacent rooms here. So the question is the meat, what we eat in these fish, is that impacted by oil exposure? And it just so happens that fish, like other vertebrates, including us, are very good at degrading the PAHs and other compounds that are found in oil. So in general, if a fish survives an oil exposure without any significant impairment to let's say, the sensory output or input and their cardiac function, with time, they're going to be fine for consumption. These compounds are transported to the liver where they're degraded and excreted. It's not the case for invertebrates, at least not to the same extent. So shrimp, oysters, and stuff, there's a different issue. But fish in the Gulf of Mexico, it's perfectly fine to eat them. So that's actually really, really hard to assess. I know some oyster banks, some oyster fishing, and some shrimp fishing, for example, has yet to recover. I know that there are many deep sea areas that are still severely impacted by oil, and part of the reason for that is that some of the oil remained at depth where temperatures are much, much lower, so degradation of the oil is slower. So there are impacts in those areas. The question about impacts to the pelagic fishery, for example, is really, really hard to address. One of the reasons, and I think it was a good idea, is that the commercial fishing in the Northern Gulf was closed after and during the oil spill for about an 8-month period. And that, of course, is to relieve pressure from the populations and also to avoid people eating oil-contaminated fish. But what that means is that you have a negative impact on the oil spill, and then you have a positive impact on the fishery closure. And the outcome of that may be a zero gain or some, but that doesn't mean that there was not an impact of the oil spill. I will say, though, and not all of my colleagues are going to agree, but I will say that looking back to what we feared in April 2010, or even through the summer of 2010, and then comparing that to what we see today, it's probably not as bad as we feared. The question is, how are we doing on mahi today? Are there as many as there used to be before the spill? Some fishermen report less, and some maybe not. There was a couple of years after the spill, there definitely was a low. It's going to be really difficult to tie that exactly to the events that happened in the Northern Gulf. And I've showed you already the migrations, the extensive migrations that these animals perform. So it's hard to say that a spill in the Northern Gulf of Mexico would impact the mahi we see off Miami. Having said that, I don't want to rule it out. I actually happen to fish for mahi as often as I can get away with. I mean, we talked earlier about this. And it's not as easy as it used to be, I'll tell you that. But I also know that over that time, the number of, say, sports fishing vessels, for example, in this area have doubled. So there's more of us out there looking for mahi. So I actually think you're going to be really hard-pressed showing an effect of the oil spill on the mahi populations today off Miami.
Thank you for coming. I really enjoyed your lecture. Have you seen any impacts on changes of the DNA of these fish? That's question number one, and also the impact on the coral reefs. What are you seeing?
Right, so I'm not going to repeat the question because you used the mic, so that should be in the adjacent room also, right? Am I correct? Okay. So 2 questions. Are there impacts on coral reefs? That was one question. And I know that there are deep water corals that are impacted in the Northern Gulf of Mexico. Absolutely. And still today, there are lingering effects in those areas. And I'm sorry, you're going to have to repeat the second question. Yeah, right. So we have not looked at direct damage to the genetic code in these animals, but others have. And in coastal species like the mummichock or the fundulus, for example, there are reports of damage to DNA due to oil exposure.
Okay. Thank you for your lecture. I'm sorry, I'm all the way over here.
Okay.
I'm volunteering at Rasmus currently with Baker's Lab. I was interested if you know the physical mechanisms of why the eye isn't as developed in the mahi-mahi and the yellowfin when exposed to, like, is it because during cellular development, maybe other organs are prioritized since they've already been under stress? Or is it just mainly because of the toxicology of the PAHs?
So that's a great question. So, and we're addressing that. Some of the gene expression work we've done actually leaves us with a quite clear picture. So retinol, which is a protein in the eye that captures the light and basically transmits that energy into a nerve signal. The formation of that protein, the biochemical pathway involved in producing that protein, is impacted in animals exposed to oil. So that's probably why they can't see. Others will argue that the impact on the eye is secondary to impacts on the heart, because if the heart is impacted, you're going to have less blood flow, blood supply to the eye, and that's going to slow development of the eye. I respectfully disagree with that view, and the reason for that is that we actually see impacts on visual acuity at concentrations that are close to an order of magnitude lower than the concentrations that induce cardiac defects. So that story just doesn't hold, respectfully. But yeah, so it's basically a specific molecular mechanism that impacts the development of key proteins in the heart.
So,
in the eye.
Sorry, just a follow-up.
Mm-hmm.
If you see visual acuity lowering at lower concentrations, then are we assuming that the eye is more sensitive to oil exposure than the heart is?
Well, I would say the development of the eye is more sensitive than development of the heart. That's what it looks like.
Okay.
But if you take the perspective of the animal, like all it takes is one organ to fail, and you're done. It doesn't really matter whether it's the heart or the eye.
Is the mahi's brain speed affected by the oil?
But that's another great question. Smart little one. So the question is whether the brain function of these fish are impacted by oil exposure. I did not touch on this data at all, but we actually have done that. We've not done it on mahi because mahi are really hard to do behavioral studies on. They don't really behave well. Either they do nothing or they eat each other. So it's hard to do those kind of studies. I'm actually not kidding, that's how it is. But we've done work on red drum, and red drum exposed to oil during embryonic development, they tend to make really poor choices later in life. So no, I know this sounds funny. I'm not kidding. Like they will, for example, they will choose risky behaviors in presence of a predator. That means you get eaten. They will choose different habitats, so a more open habitat as opposed to a habitat with cover, for example. That again means you get eaten. We've shown this for red drum. I don't have the data here, unfortunately. And interestingly, it actually correlates with studies on humans showing a lower IQ in young adults that have been exposed during embryonic development to PAHs. So yes, if you're exposed during embryonic development or as a fetus, you will have a reduced IQ and you will have impact brain function later. We didn't touch on that data today, but that is a finding that we've reported. Oh, I see. That's a great question. I'm going to repeat that question because you don't have a mic. So the question is that brief exposure, is that going to affect the animal long term? We've actually done that experiment, again, not on mahi because mahi don't play well with others, but we've done it on red drum. And on red drum, what we've seen is a 24-hour exposure will have the same impacts as we've seen on the mahi. At slightly higher concentrations, red drum are a little tougher than mahi. But if we rescue that animal and keep it under clean conditions for up to 6 weeks, we still see that impairment in ability to swim. So this damage is, if not permanent, at least long-lasting. And again, if you're a mahi in the Gulf of Mexico and you have a severe reduction in your ability to swim for 6 weeks, it's not looking good. But stay tuned. I mean, we'll know in August.
Thanks, Martin. You have 544 students.